And this is slide just to remind me this is something happening very quickly. And also we have very rich database. And I just put the 2 covers of the journal when Human Genome Draft is published. Then that's sort of like a time we are into, really into the genomics. And this has been a while, to now. So this is just remind me. And the list, the brief list is something more earlier. Because things are happening very quickly, but the basic principle or basic developments are still the same. And we modify our method based on our understanding on the molecule, on the chemical basis of DNA. So this is something important in this field. So the short history actually is just a history before all the data coming or all the data are widely available. So this is a really a very early history of this field. Okay, so if we start from, so this is a list and the last page of your handout. So just a stapling together. So the last page of the handout, we start from 1904, and that's the year we start have the using, or the first time we use, when I say we, it's someone, some scientist, use the molecular data to reconstruct phylogeny, or just to think about how to compare the molecular data among different species and try to use that to understand the relationships of the species. And we know protein sequence, we can do protein sequence pretty early. So this is in 1952. The scientists compare the protein sequence from different species. So the same thing. We want to look at how the protein change over time. What do you mean by over time? It would be more clear in a later lecture. Because what we are compared as always the modern species. Is the species living, the living species on Earth, most of the time. Occasionally, we can have some fossil DNA available. Nowadays, it's a very hot area and very informative, and it's very good. But most of the time, we only have the living species sequence available. But when I talk to you, I say we try to understand the changes over time. But specifically, the practical part, we are comparing the sequence from 2 living organisms at the present time. So we have to infer their common ancestor. We have to know or have some assumptions to reconstruct the ancient sequence. And we don't know the answer. That's the tricky part. We don't really know the answer. Nobody will tell you it's yes or wrong unless you have fossil record and the sequence from the fossil available, which is very rare. It is at present time. Not every fossil can provide good DNA sample. Okay. So you have to make an inference and to look the changes over time. So that's the important part, and that's the practice we do every day. Start from the protein. And this is something everybody knows. In 1953, we know DNA structure. We know how exactly the genetic material store the genetic information. And at that time, we already hypothesized. See, I always use we, means Watson and Crick. They made hypothesis and talk about how the information can be faithfully replicate in our life and through generation. And this is the very important part. That's why I say genetics is important, but you haven't taken the genetics, you have a genetics section in your general biology. If you do very good in your general biology, you should be okay. This is something else. We can look at electrophoresis. We can look at the protein at the population level. We have the protein sequences, but now we can look at protein. And this technique, so this is a technique, but this technique revolutionized the field. They make the molecular data more available to more people in this field. And this sounds really interesting because this is the first time we are thinking about to make a specific construction, and now we call phylogenetic tree. If I remember correctly, or I try to do, is every time when we say phylogenetics and make a phylogenetic tree, we use reconstruction, phylogenetic reconstruction. The reason is we don't know exactly. We are using, see, I repeat the same thing. We're using the available data at the present time to make our best estimates about what happened in the past. So that's what we do. And this is like in 1963, this is the first phylogenetic tree available using the molecular information. This is a big thing. We will talk about that. So we'll talk about molecular clock, like your watch. And it moves every second or every minute. So it's a constant rate, at a constant rate, right? So you know now is probably 11:40. Then if the molecule change at a constant rate, it's just like a clock. We use clock to know, oh, it's probably about the time I should quickly finish this lecture, right? Because I have to stop at 12:10. So this is what I'm trying to do. Then you are probably thinking, I'm really hungry, especially if some of you didn't have breakfast. You're really hungry now. The molecular clock is the same idea about clock. So we know time based on the clock, based on the sun, the position of the sun. So we get the idea about how time passes during the day. Then the molecular data doing the same thing or can tell us the similar story if they change at a constant rate. So we will talk about that in our probably third lecture or fourth lecture. It's like whether it's a constant rate. How do you know if it's a constant rate? And if it's a constant rate, and can we use that as information to learn more about the animal evolution or the plant evolution? So this is something, and there's a debate about it. You know, everything in biology, there's something different. So everything is different. Everyone is different in the room. So that is important. But if there's a general basis, more or less we can follow. Like we all come to the class on time and we all concentrate on the lectures. So we do something similar, but we are different. And molecules is the same. There's some basic principle they shared, like everybody, not everybody, but most of us use DNA or RNA. Now us means everything around us, the bacteria, right? Everything in the room. We use DNA or RNA as genetic material, pass on to the next generation. That's the common principle we do. And we all form based on a cell or cells. So that's something we are in common. In molecular evolution, that's the same thing. There's something in common. All the molecules do, or molecules influenced by the same principle in life. So that's something, it's very interesting. So that's why I say you have to learn a little bit biology before coming to the class. Okay, 1966. That's another thing, because we already know electrophoresis. But Lewontin and Hobby used this technique to survey the population. So at that point, most of science were really surprised the amount of variation we observed in the population, in natural population. Because since Darwin, and also before Darwin, from Lamarck, we know things are different. We know, although we all look like, or we are the human being, but we look different. So there are variations exist in the population. We have different height, different weight, different skin color, different eye color, different hair color, different, then you run out of the word, then that's it. So there's limited genetic variation exist in the population. But when we look at the molecular level, wow, it's really different. Actually, at the molecular level, there's a lot more genetic variation, and which is very interesting observation in evolution. Why? We like to ask why. We like the 2-year girl or 3 years boy. So why? Why there's so many genetic variation? And what is the mechanism, or how it becomes this variable? So, and this is starting, so all this thinking starting from 1966. And just happened that is also a revolutionary time, because the amount of data. When we look at the morphology, even you are very good, you look at the plant, you immediately spot, oh, this is something, and this is another species. You are really good, and you tell people they are look alike. Look again, they are different in the tip of their leaf, and you can tell them why it's different. But for ordinary people like me, I say, oh, they look very similar. You are not really a good observer, if I want to say so. But even we are so good, that we're still limited by what we can observe. So the technique opened a different window to let us look at the protein polymorphism in the population. So this tree become more complex, or this tree looks like more similar to what you've seen in all different papers in this field. So we construct. So they publish Fitch-Magalish, you use their method in this class. Then the constructions of phylogenetic tree. That is why I have to say we do reconstruction. Actually, there's a tree. There was a tree because we all believe species evolve from their ancestral species. But we don't exactly know each lineage and each branch. We still don't know a lot of living things on Earth. That is why we only know part of the truth, and we only have part of the data. But we try to understand how these organisms evolved during the past, say, 2 million years, 10 million years, or whatever. So this is something we do. So this is like the first, looks like a modern phylogenetic tree. And this is really important. So there's a news about this paper, and this, I forgot to say, that's a science paper. Say, wow,